E ka mana e ka reo e rau rā ki te rama. Tēnā koutou. Ko Trish Priest, tāku ingoa. Ko te manakura, te wāhaka mātou haura, te whare wānaka o tākou ahau. I'm Trish Priest. I'm the Acting Pro-Vice-Chancellor of Health Sciences. And I'm here to welcome you to Professor Gabi Dachs' Inaugural Professorial Lecture. I'm very privileged to be able to attend the Inaugural Professorial Lectures in the Division of Health Sciences. It's always exciting to learn more about the life and work of the amazing people who work for us all over the country, not just in Dunedin where I'm based. So it's just a real pleasure and privilege to be here today. Professor Dachs is going to be introduced properly by other people, but I just wanted to note that she's a research professor, which means that she has demonstrated sustained, outstanding leadership and competence in both research and service. So as well as being a medal-winning researcher, she's also contributed to the university, including through teaching, which is not necessarily usual for research professors, contributed to her discipline, her professional community and the general community through a lot of community engagement and education about her field, cancer. Her promotion is testament to her exceptional work and dedication, and I'm really looking forward to learning more about her journey as a Scatterling. I'd want to particularly welcome her husband Andy and her daughters Nikki and Laura and sons Daniel and Ryan, her friends Beverly and David, and any other family and friends who are either here or watching online. So my task is simply to welcome you and pass you on to Professor Martin Kennedy to introduce Professor Dachs properly. So again, I say welcome. Nō reira, tēnā koutou, tēnā tātou katoa.
Kia ora koutou. As Professor Priest has said, it's my pleasure it's my role to introduce Gabi and flesh out a little bit more about her before she then tells us all about her career and research and life story. So it's an honour to welcome you all here today, and it's also a pleasure to see Gabi's family in the front row there. They've shared this journey with Gabi, and I'm sure they know that very well. And I think I've seen a few of you in Gabi's office over the years, so it's nice to see you all grown up and here today. Inaugural professorial lectures are a really special occasion. As Professor Priest has said, the bar is set very high to become a research professor, and in my view, it's more like a pole vault than a high jump, and Gabi's managed to do that very effectively. I don't want to steal her thunder, but I'll tell you a little bit about her. She was born in Namibia and did her BSc Honours in microbiology at University of Cape Town in 1988, PhD in microbiology at the same university in 1992, and then moved to the UK and had a postdoctoral period for 3 years at the MRC labs in Didcot, and then moved into a scientist role at the Gray Institute, Cancer Research Institute at Northwood, not too far from London. And she spent 7 years there before joining us around about 20 years ago in the Department of Pathology, and started as a research fellow, and now here she is today, having made that final high jump, and we hope she'll be with us for quite a long time to come. I think so. Over this past 20 years, Gabi served as a co-principal investigator for the large Mackenzie Cancer Research Group, probably our largest group of cancer researchers. She's a highly collaborative researcher. She collaborates locally, nationally, and internationally, and I'm sure you'll hear a little about that. People like working with Gabi. So there's a lot of collaborators. She's also a very popular and sought-after postgraduate supervisor, and she's supervised around about 20 postgraduate students, 10 of whom are PhDs, some of those still in process, and I'm sure are here today. She's also supervised 17 summer students, which is a pretty impressive tally. She has, as you know, a sustained and outstanding level of research productivity, and we can measure that in lots of ways, but she did receive the University of Otago Christchurch Gold Medal for Research in 2021. She received the Robin Fraser Research Supporter Award in 2019, and a UOC Teaching Award, or University of Otago Christchurch Teaching Award in 2015. She's won over $5 million as principal or co-principal investigator in external grants, and has contributed to other grants to the value of about $14 million. All of that has led to her publishing 80 papers across a range of disciplines, almost all related to cancer, and her papers have been cited 4,500 times or thereabouts, which tells you how much of a mark she has made internationally. She's worked on gene therapy, effects of low oxygen or hypoxia, and vitamin C, and again, I'm sure we're going to hear more about that very shortly. One of her papers has been cited nearly 1,000 times, and she has about 10 papers that have been cited more than 100 times. So these are very impressive figures. So not only is Gabi an accomplished researcher with an international reputation, as a colleague, I'd say she's very proactive, very supportive, very positive, and she's a lot of fun. So I'm going to finish with a very brief anecdote which tells you something about Gabi. Gabi and her family, I believe, have some animals on their lifestyle block, and that includes chickens and a horse. And I can confirm, indeed, the chickens exist. And I remember through the COVID lockdown, a Zoom when this startling Gabi was looking very relaxed on a sofa, and two glorious and quite extroverted chickens marched along the back of the sofa, which caused everybody to crack up. I'm very pleased to report that the horse didn't try and do something similar. So Gabi, we look forward to your professorial lecture and hearing about your research, your career and your life journey. So over to you. Thank you.
Tēnā koutou katoa. No Namibia, ahau, kei o tatahi toko kainga e naenei. Ko Gabi Dachs toko ingoa. Ich bin in Namibia geboren. Ich wohne jetzt aber in Christchurch. Mein Name ist Gabi Dachs. Goeiemorgen. Ek is in Namibia gebore, maar ek woon nou in Christchurch en my naam is Gabi Dachs. No reira, tēnā koto, tēnā koto, tēnā kato katoa. Kia ora. I'd like to start off with acknowledging this beautiful korowai. Thank you, Suze, and thank you, Auntie Marahu. That is beautiful. I feel very privileged to be wearing this. So you might have noticed the music on your way in. It's a song that is close to my heart. We had good times with it in Cape Town as undergrad students in a very crowded, low-ceilinged, stuffy students' union, bopping up and down with live music in front of us. So it brings back memories, but it also is quite relevant to my journey through research, and also a journey from leaving Africa. It is about displaced people from Africa, and it's originally obviously for the black South Africans during apartheid, but it is it's more general than that, because humanity has started in Africa, and the song itself makes reference to the Alduva Gorge in Tanzania, where supposedly the first human remains are from. So hence the scatterings of Africa. So I give you a moment to reorientate yourself. This is the globe with New Zealand firmly in the center. So as Martin has already pointed out, I was born in Namibia, then moved to Cape Town in South Africa, and then did postdoc in the UK, and then 20 years ago we came here. So I thought I'd give you a little bit of a background where I'm from, what my whakapapa is. It's German. Although my grandfather on my mum's side was born in St. Petersburg. I think they were only there for a short period of time and then returned to Germany. But he then emigrated to what was then German Southwest Africa, and they worked in a diamond mining town called Kolmanskuppe on the southern border of Namibia, where he met his wife, also a German immigrant. And my mother was born in Kolmanskuppe, so that is these houses here. It is now a ghost town, and it's very popular with photographers and tourists because the Namib Desert is taking over the area. My father and family are from Germany. When he was 18, he joined the Wehrmacht in 1944 and got sent east into Russia, where he was captured and spent 5 years as a prisoner of war. Following that, so this is my father here, he and his brothers left Germany and came to Southwest Africa. So what you can see, this strange looking concoction in the middle here, is an indication of what Namibia was then like. It was like the 5th province of South Africa, and it had alongside of it the apartheid laws and all of that goes with it. So this is a picture of Namibia. Not all of it obviously looks like this. This is Etosha Pan up in the north. Area-wise, it's a little bit smaller than Canterbury, but not much. It's a huge area, so Etosha Pan, the wildlife park. Namibia itself is 3 times the size of New Zealand, and it's got about half the population, so it's pretty sparsely populated. So this is the outline of Namibia. So this here is the coastline along here, and the coastline has about 70 kilometers of pure desert, which I thought until I was an adult that all coastlines looked like that, and that you don't have green stuff growing anywhere near the coast. Windhoek is right in the center here, that's where I was born, and Etosha Pan is up here. So what got me into science? Well, I was always into animals and biology, and then slightly more recently into chemistry. So following 13 years at school with Abitur as the school send-off, I went to Cape Town, the University of Cape Town. At that point, Namibia did not have any universities, so virtually everybody who went for tertiary education went to South Africa, one of the universities there. My brother and I started university the same year, and we were the first in our whānau to get tertiary education. My mum was a bookkeeper, my dad was a carpenter, who then also worked as a builder. And as I said, Namibia was the 5th province of South Africa, so we had apartheid. It was quite a traditional conservative country, although in the last year of school our eyes were opened somewhat as to what was happening in the world. But certainly the first few years at UCT opened my eyes a bit more. We had, UCT was one of the centers of the anti-apartheid movement. So we had police and army coming onto campus quite regularly. This is a picture which I actually recall very clearly, where they came onto campus and tear gas was fired into the library. So we made a very hasty exit at that point and left. So it was certainly interesting times, and it widened my eyes to what's happening in the world. So onto a bit more science now. So I did my PhD at UCT, as was said. I'm not going to go through with you exactly what my PhD was about, because I can't remember most of it. But it started my love for looking at low oxygen conditions. So I was working with an obligate anaerobe called Bacteroides fragilis, which is very important medically, because if you have a infection with Bacteroides, it can be very, very severe, and it gets usually treated with a drug called metronidazole, which only works under low oxygen conditions. So these anaerobic bacteria find oxygen is toxic to them. So this strange looking beast on the bottom right here was the anaerobic chamber that we used and that I used as a student. So you came into the room and usually had the arms out because there was internal pressure of gas sticking out. The atmosphere in there was controlled with a palladium catalyst and hydrogen, and that removes any trace of oxygen. But I also found out that if the hydrogen wasn't regulated to the right level and somebody opened the door and it reacted with the oxygen of the atmosphere, there was a flash. It was quite scary. So this piece of kit was actually the anaerobic chamber was the first thing that I bought, or that we bought when I came to the UK. So a quick thing about oxygen levels. As I said, this is my great fascination. And the bottom, that long graph here shows in a log scale oxygen concentrations in the atmosphere, or often it's calculated out because we're working in tissues as well. So the air that we breathe around here has about 21% oxygen. And then the pulmonary vein in the body is around about 13% oxygen, so it's a bit lower. And then all the different organs in the body have different levels of oxygen in them. So kidney, for example, has a range of about 2 to 9. Calculated out, uterus is quite low. And then when we come further down, most tumors have areas of very low oxygen, and they can range from 0.1 to 1%, and even areas with no oxygen containing at all. And then the anaerobic bacteria I was talking about, Bacteroides fragilis, needs 0% oxygen in order to survive. So I'm working, my whole career really was centered around this area, and I'll be telling you a bit more about it. So as I said, as a postdoc, Andy and I moved to the UK without a job at first. We found our jobs when we were over there, and it was quite hair-raising. So this beautiful picture here is the Didcot power station, and this is near where I worked, the MRC at Harwell. Usually when MRC Harwell had any sort of conferences on, they put pictures of Oxford and the spires up. Not this. So this was my postdoc research. How do human cells sense and respond to low oxygen levels? Why is that important? First of all, I'll start with that, because patients with hypoxic tumors have a poor survival. So this is not my work. This is was done before me. And this is an example here showing how that work was done. Basically, there's an oxygen electrode that can be inserted into tumors in patients, and oxygen concentrations are measured in a stepwise fashion throughout the tissue. And from that data, these graphs here were created. So on the y-axis, we've got the percentage overall survival, and then on the x-axis, the follow-up in years. And what this shows is that tumors that have less hypoxic areas, those patients actually fared better compared to the ones that had more hypoxic tumors. And that's why looking at hypoxia in cancer is really, really important. So how does it arise? So tumors, because they grow fast and because they have an aberrant kind of growth factor stimulus, often have inadequate blood supply. And in this picture here, again, it's not mine, you can see like areas where you can't see any of the vessels. This is a vessel cast. So vessels were infused with a reagent that could then solidify. And at the same time, because tumor cells grow rapidly and they consume a large amount of oxygen, the oxygen goes down quite rapidly within tumors. So that then results, and this is a beautiful picture from my student Linda Bus, showing in picture form, this is a section through a tumor, and you can see in green the areas that are hypoxic, in blue areas that are perfused, and in red where the blood vessels are. So you can see that around the blood vessels, by and large, you have areas that are well perfused, well oxygenated, whereas at a distance, you have areas that are very much viable. These are very much alive, these cells, but they are low in oxygen. And that then means they upregulate cell survival pathways, and one of the important factors in those cell survival pathways is the hypoxia-inducible factor, which is very close to my heart, and I'll tell you a bit more about it. So in my postdoc research, this was the area I was particularly looking at, the hypoxia-inducible factor, and it controls gene expression. So here's just a picture of a DNA strand, and what HIF, which is what I'm going to call it from now on, does, it binds to it and then increases expression of the genes that are adjacent to that. So the way that we kind of proved and confirmed that is we had the area that the HIF factor binds to. There's a short sequence of DNA that it recognizes called the hypoxia responsive element. We had that adjacent to a reporter. So when HIF bound to that short sequence, the reporter gene was expressed, and we could find that and measure it. So what this picture on the right here shows is that as oxygen levels go down, so we have normal oxygen concentrations here going right down to anoxia versus fold increase in gene expression, the more hypoxic the cells were, the more we have that reporter gene coming up. And interestingly, there's a difference even between just gassing with nitrogen and absolute anoxia. So this system is really sensitive for very, very low oxygen concentrations. So what? It's really important for tumor growth. So this we showed by implanting mouse tumors into mice. So we had one set of tumors which were just normal wild type. They had everything that a normal tumor cells have. And then we had one where HIF was mutated, so the HIF was not functional. This survival pathway could not kick in when the tumors found themselves under low oxygen. And then when you looked at the growth of these tumors, on the left-hand side here, we've got the ones that are able to respond to low oxygen, and the tumors grew rapidly in these animals. And when the HIF was mutated, they grew much slower. So if you think about that as a possibility for patients, if we can slow down tumor growth, that would be an advantage. So this area of research has made quite a lot of headlines. The person that we collaborated with in the UK, Peter Ratcliffe, is one of the 3 recipients of the Nobel Prize a few years ago for finding out exactly how cells respond and sense oxygen, and my work's played some minor role in that discovery. So this is how it works. Sorry, this is going to be a little bit heavy. This is a bit of science coming up. So what this work has discovered is that there are enzymes. These enzymes are catalysts that do things in cells. So we just call them PhD and FIH. And what they require is they require oxygen in order to function. They require a bunch of other things, which I'll also be talking about. So when these enzymes are active, they take the HIF, which I've already talked about, and modify it. They hydroxylate it. And when HIF is hydroxylated, it gets set on a path to destruction. So in normoxic cells, in cells that have normal amount of oxygen, HIF is destroyed and cannot function. However, if you have low oxygen conditions, these enzymes are not active, HIF builds up and then activates a huge number of pathways, survival pathways that allow tumors to grow, to spread, to grow their own blood supply, but they also are involved in resistance to therapies like chemotherapy, radiation therapy. So when HIF is up, these tumors are aggressive. And that is all because these enzymes up here require oxygen to function. So I'm leaving out a big chunk here, which basically don't fit into the story. I'll stick with the hypoxic theme. So in 2003, we decided to immigrate to Otatahi. We came here for a visit, and we found that it's a beautiful, beautiful country with friendly people. And I could see that we could have a work-life balance in this country, which was really hard in the UK, where you spend at least 2 hours a day in traffic trying to get backwards and forwards. So one thing that we did quite recently, in hindsight, we should have done that much, much earlier. We did a survey in patients with cancer. And we asked them what would they like us to do research on. What's important to people that live with cancer? Mate puku puku, he aho o whakaro. What's important? So we questioned 204 participants, and they included both patients with cancer and their whānau. And we asked them a range of questions. What we found is that virtually all of them found that cancer research as a whole is really important, so high or very high priority. We gave them a long list of keywords to choose from, which they found, they had to tick which ones were important. And cancer screening, quality of life, and Māori health were keywords chosen most often by the participants. We also asked them to write down in their own words what we should do regarding cancer research. And it became very interesting when we looked by ethnicity. So we had enough Māori participants to draw some conclusions from this. And interesting, Māori participants were very interested in getting answers to their questions regarding complementary and alternative and holistic therapy, as well as access to care. Non-Māori participants were very interested in cancer risks and causes and early detection and screening. All of these are areas that are very important and that we actually work in the Mackenzie Cancer Research Group. So complementary and alternative medicine include, for example, vitamin C. And here we've got a beautiful picture of vitamin C under polarized light. It is a beautiful, small, tiny little molecule. So why am I interested in vitamin C? How does that fit in the story? You might not have spotted this, but one of the cofactors that's required in these enzymes that can sense low oxygen is vitamin C. Without vitamin C, the enzymes will grind to a halt because the ferrous ion in the active center becomes oxidized and they stop functioning. So if we have vitamin C coming in, they can get turned over and the system continues working. So why do we need vitamin C? As this is a quick overview for those who don't know. Most animals can make their own vitamin C. So your rabbit and your horse and your chicken, they can all make their own vitamin C, usually in the liver. But we, together with a few other animals, have got a mutation in that pathway. For example, guinea pigs, humans, and zebrafish, we all need vitamin C from our diet. So we measured vitamin C in cohorts of patients with cancer, and the outcome was quite interesting. So we had, here this is work done by summer students over several years. These summer students were medical students, so they were fantastic at it. And they recruited patients that were either pre-surgical, so early in their cancer journey, and they were about to have their tumor removed, or they were further along the pathway, so they were receiving chemotherapy. So they were recruited in the Christchurch Hospital chemo suite, or they were getting one of the newer types of targeted therapies, immunotherapy. And it's interesting to see that we're looking at vitamin C on the y-axis here, so healthy vitamin C levels in your blood are around about 70, 80 micromolar, up to 100 micromolar. You can see these individual dots here, or all individual patients. There's a huge range in all of these three cohorts. But if you look at the little red line, that's the median, so the midpoint. And with these increasing more complex treatments, the vitamin C in these patients was lower and lower and lower. And if you look also at the bottom here, for example, the immunotherapy patients, they have a large group of patients that are very low in their vitamin C in their circulation. And these patients are probably en route to becoming scorbutic. So we were interested not looking at in blood, but we wanted to see what actually happens in the tumor. And we have a marvelous treasure here in Christchurch, the Cancer Society Tissue Bank. And this is just showing how we get tissue samples that we can use for research. So the cancer is removed in surgery by the surgeons, and it gets sent to the anatomical pathology, where a pathologist is able to select a piece of tumor that is surplus to diagnostic requirements, because we do not want to interfere with anything like that. The example here shown is a bowel cancer with the normal mucosa around it and the tumor in the center here. And the tissue bank only needs a tiny little sample, and we need an even tinier sample, so it's the size of a pinprick shown here. In order to measure vitamin C in these tumors, they get ground to a fine powder in liquid nitrogen under very, very low temperature. And then a machine called an HPLC can exactly measure the vitamin C in that tumor sample. And this is an example of a peak here. This is the gold standard used for measuring this very unstable small molecule. So what did we find out? We have looked at numerous types of tumor. We looked at brain cancer, breast cancer, kidney, bowel, and cancer of the uterus, using the He Taonga Tapu Cancer Society Tissue Bank samples. And we had about somewhere between 50 and 100 samples from 50 to 100 patients in each of these types of cancer. And I will just summarize what we found, showing brain cancer here as an example. So on the y-axis is the amount of vitamin C, or ascorbate, that we measured in the tissue. And here we compared tumors that were either low-grade or high-grade. High-grade tumors are more aggressive. They tend to metastasize more easily. And we found that the tumors that were more aggressive had lower levels of vitamin C. So that could definitely be of clinical significance. We also, as you can see again from the individual dots, that it varies hugely between patients. And what I'm not showing here, we also looked at normal tissue, not in brain cancer, but in some of the other types of cancers where we could get a normal tissue sample. And it was not predictive of what happened in the tumor. And we believe it's because the tumor has this really abnormal vasculature and poor diffusion that it behaves differently from normal tissue with regards to vitamin C distribution. Because I'm still very much interested in the hypoxic pathway, we could also measure the hypoxic pathway in these tissue samples, in the exact same ones that we measured ascorbate in, or vitamin C in. And we could find that, we could show that the ones that had low vitamin C had the more aggressive, higher levels of the HIF pathway, the hypoxic pathway. And this is exactly as we expected. When you have not enough vitamin C to turn over the activity of those enzymes, the hypoxic pathway is able to come up and activate. And then those brain tumors, in this case, with higher vitamin C, had lower levels of the HIF pathway. could that be important? So we are able to do association studies with these samples because we have ability to do a follow-up on the patients. So in this case, again, I'm sticking with the brain cancers, which is the most recent work we've done in glioblastomas, which is a very aggressive type of cancer. Here again, we're looking at the probability of survival on the y-axis versus the years follow-up. So patients with a low HIF score, an indication of a less aggressive tumor, had slightly better survival if you look at the green curve here, and the ones that are high HIF score had a poorer survival. They don't separate hugely, but they do separate, and it's a good indication that this could be important. Then we also did the same thing with vitamin C. So the patients that had low vitamin C in the tumors, this is not circulating vitamin C, it's in the tumor, in the red here, had really poor survival. The ones with higher vitamin C had slightly better survival. So they lived slightly longer as far as the glioblastoma tumors go. We've also found similar things when we looked at patients with breast cancer or with bowel cancer. So there is something happening, but this is an association, this is not a proof of causation. So this is one of my favorite graphs of all time. This is an association between the chocolate consumption in a country versus the number of Nobel Prizes that country has won. So the countries that consume a lot of chocolate, like Switzerland, have a lot of Nobel Prizes coming their way. So one may draw your own conclusion, but no, it's obviously not a causal relationship. So if we want to work any further with the vitamin C story, we need to look at if we change vitamin C, does it actually make a difference? And we've completed one small clinical trial, and we are about halfway through another small clinical trial, where we're looking at whether if we increase the amount of vitamin C in a tumor, can that make a difference to the biology, at least, of that tumor. So this study was done in patients with bowel cancer, where we got a tiny little sample during colonoscopy, and then the patients were randomized into either a treatment group or a control group. The treatment group received 4 days worth of high-dose vitamin C infusion, 75 grams per day. This is a really seriously high dose infusion, which is what's normally used by the alternative practitioners out in the community. And then we got a sample at resection, and we could measure both the ascorbate, the vitamin C, before and after, and we could also look at the HIF pathway. So this is just shown here in a pictorial form. So this is an example of a bowel tumor with the background here, the normal mucosa, which is the normal tissue. And at biopsy, the tumor and the mucosa were of similar vitamin C concentration. These were actually measured. I'm just showing it here in color. And at surgery, we could sample across the tumor from the outside of the tumor towards the center. And we could show that the center, which is poorly vascularized, also has the lowest vitamin C, which makes sense because vitamin C gets consumed and it doesn't enter the area which has a poor vasculature. The patients that received infusion looked similar at the biopsy stage, but when they had the 4 days of high-dose infusion, we could increase vitamin C concentrations, but very much still there was that gradient across the tumor, with the outside of the tumor getting more versus the inside of the tumor getting less. Here we could show that with the infusion, on the right-hand side here, N being the normal mucosal tissue is very low, but we can increase the amount of vitamin C across from the outside to the center. And the amount of the HIF score followed the same pathway. So this is exactly as we predicted. So as a summary, my many years of work has shown that hypoxia drives a complex survival pathway in tumors that enables cells to grow and to avoid therapy. Vitamin C is low in many patients with cancer, and also in tumors, and specifically parts of the tumor that are low in oxygen as well. And that enables an aberrant hypoxic pathway to be upregulated. We've shown that high-dose vitamin C infusion may dampen the hypoxic response. We do not know yet whether that has any effect on survival of these patients. They haven't been treated for long enough or followed up for long enough. So what did I learn? It's absolutely vital to have a close relationship with clinicians. That means we can answer the relevant clinical questions. Research is important. We don't know everything. A lot of people think that we know everything there is to know about vitamin C, but we do not. And that people that live with cancer deserve evidence-based guidelines for their informed choices, and that's what we as researchers are here to do. And that includes things like complementary medicine, such as vitamin C. The picture on the right here, I've asked my niece whether I can show this. This is my niece's little boy who's currently undergoing treatment for leukemia. And it's people like that that we do the research for, that we need to get answers to them about fundamental questions, and that might be of importance. So I'd like to end up with my special thanks. Specific thanks to patients who participated in our clinical trials so far. You can see on the left here, this is one of our participants getting high-dose vitamin C infusion. Also the participants in all our other clinical studies who answered questions, who provided blood samples, thank you very much. Patients who, with cancer who have gifted samples to Taonga Tapu Cancer Society Tissue Bank. Without them, we could not do this research. I'd like to thank the nurses and staff at Christchurch Hospital, especially those in the chemo suite, the endoscopy unit, and the obs and gynae unit, and the nurses and staff at Christchurch Clinical Trials Unit across the road who have done the high-dose vitamin C studies with us. I will not be able to mention each and every person who was involved in this work, so I'll go through by groups. Obviously, there are a lot of fantastic collaborators. So I'd like to first of all thank the highly talented and fantastic students that were involved, the members of my research group, the Mackenzie Cancer Research Group, our clinical colleagues that have been involved, my absolutely fabulous Maori advisors, Ngāmihi, my collaborators far and wide, close by as well as overseas. And I don't want to leave out our support staff, and some of them are so good at not having their photos online that I had to put names down. I'm very grateful to the Mackenzie Charitable Foundation, who have been supporting our group and thereby me since 2010, and also the funding support from a range of other external funding bodies across the country. They make it possible that we are here and we are doing research. And I want to end up with what is our greatest challenge, and that's science funding. That is a challenge for us all, because we need our salaries, we need our research funding. And finally, my exceptional mentors throughout the years. And I will start with a teacher, my biology teacher at high school, Hugo Bovang, who got me into science and into biology in the first place. David Woods, who was a fantastic lecturer at UCT. My supervisor, Vala Bratt, at UCT. my postdoc mentor, Ian Stratford, who pushed me into giving a keynote lecture a year after starting my postdoc, which was terrifying. Bridget, my clinical mentor, thank you very much. You've taught me so much. And Margriet Vissers, who's basically my life coach in all things and who I can go through with all my questions. Thank you, Margriet, and thank you, Evan. And of course, my fabulous and amazing family. Thank you very much.
Tēnā koutou katoa. I think that's working. Yep, great. For those who don't know me, my name's David Murdoch. I'm the Vice-Chancellor of the University of Otago, and it's an absolute pleasure to be here today, to be back on campus, but also to be here at Gabi's inaugural professorial lecture. Gabi and I, we've just worked out, go back 20 years. So I was the head of the Department of Pathology. It was actually my first full year in the job when Gabi arrived, and I do remember news ahead of this hotshot young cancer researcher arriving and asking for my support to appoint her to a role. Remember that very well. And I'm so glad about the music. I picked the music. It just had to be Johnny Clegg, and that was just wonderful. I was so happy to hear that. Gabi, you've really just demonstrated, an extraordinary journey. I've learned a bit more about you that I didn't know. I knew about your research, and it's just wonderful to see it all outlined in front of us all. I mean, that's just been a fantastic journey. And I thought what I could do is just add a few more things that may not be so apparent from your talk. And some of them actually have been mentioned, and I think the first is just you've done this through a really tough environment of research funding. You alluded to it at the end, that was the only time, but actually that's and an environment that's doesn't always treat full-time researchers very well, and you've really navigated that very successfully. But I know that's not easy. That's not easy at all. You're a really superb. Your reputation as a mentor and a supervisor, student supervisor, really very, very powerful, and that's something that I really want to underscore. Your positive contribution. Martin mentioned it. Your positive contribution to this campus and the university. And your thoughtfulness and kindness. And I'm actually going to disclose something publicly that I said I'd never do. So some of you will remember this. 2019, a few weeks after the dreadful mosque shootings, just before Easter, people came to work here. And the whole building, every office, there was chocolate. In front of every door. If the door was closed, there was chocolate outside. If it was open, all the desks had chocolate on them. And that wasn't all. There were posters put up in places of two people, one dressed as a chicken and one as a rabbit, and with sort of holding chocolate. And this was just posted around in the lifts. And as Dean at the time had access to the CCTV footage and the swipe card access at certain times. And so Melissa Payton and I were able to fairly rapidly identify likely culprits. And by the way, they were waving at the CCTV cameras. And I might say that I think both of those people are here in the audience today, and one of them may have been the speaker. And why do you say why I haven't disclosed it before? Well, I think discovering who they were, then knowing who they were, these were people who just did not want to draw attention to themselves personally. That was not important. And I knew that was the case. And so Melissa and I just stayed mum, although it may have leaked out in some area. But it was it wasn't a random, it was a deliberate act of kindness that was just, at that time, what a difference it made to this campus. So I just as acknowledge Gabi and her co-conspirator, who will remain anonymous but might be able to be worked out. So thank you, Gabi, for this wonderful lecture. Really, really thank you. And I just want to have two other tasks. One, to thank you all for coming. Really important that you're all here, and it's just fantastic, and have the family and friends and colleagues, and just wonderful. And also to invite you just to join us in the foyer for some light refreshments, and we can continue the discussions. But I just before we do that, and I'll get Gabi to lead us out, I think another round of applause, and thank you for joining.